USDA, just like the other one. Treat it gently because it's $35,000 worth, okay? And some of the trays that we're going to learn how to use here, each one of these is about $800 to replace if you drop it and break it. I think it is glass of a sort inside the middle there, so I don't think you really want to drop them.
So will you do USDA replace those then when we drop them?
No,
Oh.
you have to take them. If you break, you drop, you break, you buy. Yeah. So so we need to be careful with that stuff. So I'm officially the administrator on this. This is a different system from that old one in that it has its own computer on board and it's a touch program. Okay, so everything is done by touch screen here. It is connected to a computer here. I connected it, and eventually I'll get them talking to each other, but I haven't figured out the shared folder thing yet. One of you computer gurus can probably help me with that, although it is a PC though, so I don't know. But yeah, what I wanted, the reason I wanted to connect them was because this has the touch software. It's called ImageLab Touch, and that software isn't like the full version of ImageLab. It's just to get your, to get your image made. Over here, the full ImageLab software is on here now, and you can do more manipulations with your gels. You can create a better resolution picture if you want to actually directly publish it. You can label it. You can estimate molecular weights from your markers. For chemiluminescence, you can actually quantitate the luminescent signals, things like that. I'm not going to go over chemiluminescence at all today. That's a whole other ball game on here, and I don't know if anyone's doing it right now, so we'll tackle that when somebody decides they want to run a chemiluminescent blot or something. And maybe me first, who knows. So we won't do that, but it is capable of doing that. So it does DNA and protein gel visualization. It'll do colorimetric Western blotting applications like silver staining and Kumasi blue. Pretty much that's the whole gamut really. And it has two light sources in it. It's got a UV light source and an Epi white reflective light source in it. Okay. Let's see. I do have the ImageWare software that I put on here on my USB here, and so if for some reason you'd like to have it on yours, if you do plan on doing a lot of analysis, we can you can download it off of my USB, the full software. I got it off the website. I just downloaded it, so I think it's working. And then I wanted to show you some of the extra supplies. So this drawer belongs to him. And there is the manual in here, and the manual is helpful and it is readable. So if you get stuck on something, you can look up the section for it, and it's a full printed manual. There's another sample tray in here. This is used for protein gels, and so if anyone's not doing them, no one's doing them right now. It is here when we need it for colorimetric applications. There's extra film for the thermal printer camera there. And it's pretty much the same camera as here, printer as here. It's just a newer model. And then there's some extra paper towels here for drying these trays off. Okay, and so that's in there. Okay. All right, so the instrument itself is pretty easy to use. I'm going to tell you as much as I think you can do today. I mean, it's not complicated. The software is very user-friendly, but I want you to be able to use the full breadth of it. We were never able to use the full instrumentation on this because we couldn't get into the full major software area because we had computer issues, an old computer on there. So this one will do some more, and you can manipulate your image a little bit more to get the best picture you can. Okay, so it's just a matter of turning it on. Now, I don't know what you guys think. I don't know that I want this on all the time because of the screen on it and everything. I don't know though that I don't know how many times a day it's used. I mean, if it's only used twice a day, the user can turn it off. I mean, it's just a press of a button and you turn it off, and it doesn't take much to start it. I mean, it's started now, it just took 10 seconds. And so, I don't know, I guess right now, I'd kind of like it turned off, and if it just becomes just a hassle for some reason, then we'll keep it on during the day and try and turn it off at least on the weekends or nights. I mean, there's no point in letting it sit here like this, running. I'm sure it'll shorten its lifetime if
Does
we
it
do
have
that.
a sleep mode?
I don't think so. I didn't see anything about a sleep mode. I can read a little more about that. I don't know if the screen blackens or not. I haven't had it on very long yet.
Maybe keep track the first few weeks to see
Yeah.
how much it gets used.
Yeah, I mean, we don't have a sign in though, so it's hard to know.
But I mean, like just like a checklist. I'll use it.
Yeah, we could do that. But it isn't hard to get started, and we could probably leave the printer on, but if you do find that the printer is not on, there's just a simple flip switch in the back here. Okay, so this is the welcome screen, and the welcome screen is actually not just blah 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 from the manufacturer. It's something that I wrote on there for you to read. And it's mostly just kind of comments on getting started and a few warnings. First warning, of course, involves the use of no ethidium bromide on this instrument. Now that also means that if your lab hasn't totally converted over, like the lab I'm in right now, you know, if somebody's used a buffer in a gel box and they ran an ethidium gel in it, you don't want to put your non-ethidium gel running into that buffer, because then you'll have ethidium in your gel, obviously. So you got to be a little careful if you are going to be a mixed use lab. I can't stop you from doing it, but I'm hoping we can keep it clean.
Honor
And if you do, yeah, honor system. If you do want to use ethidium still, fine. There's your instrument. Okay. All right. So I've put my name and my contact information from my other lab that I'm in right now in Christensen on here. If someone needs to call, if they need help, if they want me to train them individually, if you guys don't have time or don't want to, then they can call me, or if there's a problem, they can call me or email me. So my email's on there. This instrument is a little different than the other one in that it does use these imaging trays. And there's a couple different flavors of them we'll talk about. For imaging, you don't want to put your gels like that one directly on the light box. Nothing goes directly on this light box in here, okay? I don't know how waterproof it is. I'm hoping it is. Let's see. And then there's a little line in here about creating your username and your password. I am going to require a password. I don't think you're going to have to enter your username each time. When we see the next screen, you'll see that it's from putting them on the screen as I add them on. So I think you'll just be able to find it and press it and then add your password. Now the password protect is partly for your own security. I mean, nobody is really usually bugging with anybody else's images, but it won't allow anyone else to get into your data. Okay, and it can be a fairly simple password. There's like 6 characters or something, and with a few specials, and we'll get to that. It'll tell you on the screen. And I wanted it in a certain format, the username, so that I know who you are and where to find you if I want to start wiping images off, if we've got like way too many and they've just been there forever.
So you want each individual to do one and not like one for the lab?
You can do one for the lab if everyone wants to put all their pictures together.
Okay.
You know,
it's probably,
I would put an individual
just
one
easier,
personally.
yeah.
So that you took those shots if you want to go back to them, and you took those shots and they're not all mixed up, because you can, over time you'll have quite a few pictures.
Right. Sure.
So I would keep them separate. So what I did, for instance, with mine, it's Sue M. So it's my first name, my last initial, .Samick, because I'm in the Samick lab right now. Okay, so I know who you are and what lab you belong to. Okay, it's pretty simple, and you can abbreviate Muehlbauer if you want. Mule. And so that's just for me to manage the system, and then the username, the password is up to you, obviously. I just a warning to gently wash the trays with water when you're done. I've got a squirt bottle here. We've got the paper towels. You can either put it under the sink and wash it off, or you can just squirt it and rub it off. I do prefer, though, that you probably take this tray off of the light box to do that, because I'm afraid if people are pushing down too hard on the drawer that pulls out, that we might screw up the hinges on it. So I'd prefer that you did those manipulations off of the light box.
Okay, so the images that is it getting stored on the instrument, or is it getting stored on the?
Here, but
so how much space are we talking
It's
about?
huge, like 120 gigabytes or something. It's a big computer. So it's not going to fill up quickly.
Right.
So we can store a lot of images. Yeah. guess. I'm not, too worried about that. But, as the years go by, and it's like, who was that? And who did they belong to,
Yeah.
And it's like, we had that with the other one.
And that'll be, that's kind of the issue. That's why I asked about having each lab do one for their group, is because sometimes after people go away, who's going to be, how
I
can
can
we
get into it.
recover their password?
I can go in
Oh,
as an administrator
okay,
and get
just-
into their account.
Okay, all right.
I mean, you could set up a Muehlbauer lab, but I don't think there's any way to set up folders.
No, there
you know,
isn't. This
within-
doesn't have a
right,
folder
So I
system
mean, you
in
could
it
set
that
up
I have
Muehlbauer,
found.
but then you'd have to give the password to everybody in your lab.
Right,
Yeah.
and
Which
like
is
you said,
okay.
it's going to get so confusing if you've got
Yeah.
a jumble of images.
And
in
right
there.
now, the way the images, they have a certain format for the way the images are named. You can change that certainly and put your whole own name on it. But right now it's like, your username and the date and the time or something like that. And so if everyone keeps doing that, it's all Muehlbauer and a date and a time, you won't, which one is yours?
Yeah, exactly.
Ugh.
Yeah.
So yeah, I think
As long as you can still delete people after
yeah,
they've
as an
left
administrator, you can go in and
without having their password.
route everyone out.
That's the only thing I was worried
Yeah.
about, is if people left and,
Yeah, no.
and didn't give us their password.
No,
then-
I can ask them to change their password if I want, or I
Right,
can do
okay.
all that.
Okay,
Yeah. So
all right.
that's all right. Yep.
So right now the images are only going to be stored on the computer and the machine, and they are not. It's not hooked up to this one yet.
It is, but I haven't quite gotten the shared folder figured out
Okay.
to bring them over. And the only reason to bring them over right now is to work on them a little further, because you can print them out here, or if you prefer, you can put it on USB and print it out on another computer that can read a JPEG or a TIFF, which is everybody. So. Yeah. So, and you can't, even if I can't get this set up right away, if you want to use this other software, you can take your file off in the correct format here and stick it in there and do the analysis. I'll figure this out here with some other computer expert that's not me, and then we'll get going on that. Okay, so let's see. I want to make sure I made all the points I wanted to here. Okay, let's just start it. So, read my screen if you need some information or wondering things, if you need to contact me, but otherwise, you tap and you're into the login screen. Now, so far we've got 3 users in. Adrian, I put in one for Mindy because I was doing a tester, and then I'm here. But why don't one of you create a user?
Sure.
Okay. Okay, and then think of something quick. No.
Stop filming her.
Yeah. Yeah, 6 to 32. Oh, you need some more. You need uppercase, lowercase, one letter, 3 of those, I think.
Okay.
Or they need, it must contain all of those things, sorry.
Okay.
Ooh, it's really pretty.
Up, lower, and
Upper,
One,
lower, number, special
symbol?
characters.
Okay. What?
Yikes. No, I had to answer 4. Oh, here he comes.
I'll be here.
You've been here, yeah. Better late than never, dear. We're creating usernames and passwords or some of them.
Oh, so you have to have an uppercase and lowercase?
Yeah.
You do like 1, 2, 3?
No.
it's too bad they don't let you say this is the amount of complexity I want to.
Now, if this
require.
if you guys decide this becomes too much of a hassle, we don't really have to. This will keep anyone else out of your account. Otherwise, they can just hit your user number or your username and go into your account.
And start storing stuff in there, and then you come back
Okay,
and you don't
so-
know where it came
Yeah,
from.
right.
Yeah.
So then it gives you a message after you've gotten started, how to get started looking for an image. Now we can talk about tray choices. So there's 3 trays, 1 of which came with the instrument, 2 of which I bought. The one that comes with the instrument is this one, and it's called a Blot UV stain-free tray. And this is kind of an all-purpose tray. This is UV light transmissible. Any of these new DNA safe stains will be able to be, I think, pretty well used on this one. This, however, will allow UV light to come through, so it will damage DNA. So if you have DNA that you don't want used with UV light, you'd want to use the blue tray, but you have to pick the appropriate stain. Okay, because not all stains, like the stain I'm using right now, you can't see on the blue tray. You can only see on this tray with UV light. Okay, so you'd have to pick the right tray. And also you use this for all blots and stain-free protein gels, which are a whole other flavor of things. I don't think anyone's doing those now. And then this is the blue tray, and you can see it's clearly blue. And this one, again, is used with a lot of the cyber, green, safe, and gold dyes. And you can also use it with something called Gel Star and fluorescein and gel green. And gel green is one of the pamphlets.
So each tray, it lists the dyes.
It kind of lists them generally, but for instance, mine's called Easy Vision. Did you hear that on there? No.
Yeah.
So when we first started doing it, we had to figure out which tray for me to use.
So it's just a matter of if you get something that's not listed on there, you just have to kind of
play with a few of the things here.
Play with it and see
either you see the image or you don't.
Well, you should
so yes or no.
Yeah.
If you take a list of what it can use, you should be able to look at the emissions and everything
right.
and figure out if you're just
right. I mean, basically all of them should be able to be seen on that blot UV stain-free tray, and only some can be seen on the blue. That's really the difference.
Yeah, that's actually a good way to do it.
Yeah.
So start with the UV.
Start with the UV one, but if you don't want your stuff exposed to UV, try the blue one. But you can read in these pamphlets, it'll tell you their emissions, and it'll tell you whether you can use a blue transilluminator or not with these. And part of the other advantage of the blue, again, you don't damage your DNA with UV light exposure, and when you're trying to cut out a band, you don't have to have all the cloaking devices on to cover you. But in order to see the bands on a blue tray, you have to have gold goggles, and they're supposed to be sending me some. They didn't. I complained to Bio-Rad. I said, we spent $35,000 on this instrument, and you couldn't give me a $24 pair of goggles. So they're sending supposedly.
I'm like, I have a pair. I can bring mine up.
Do you?
Now.
Okay, yeah, because you can't see the eyes, the bands visually.
Just the amber.
Yeah, like an amber.
Oh,
I think the amber orange somewhere in that.
They're pretty orange looking.
Yeah, they're pretty orange looking.
We might have one next door too. I'll have
to
check.
I mean, because the bands I think are green, and so in order to see them on the blue, you have to change the spectrum. Yeah. Okay.
But that won't work for the photograph. To print it.
Yeah. I mean, you'll see the image and it prints. Yeah.
Okay. I mean, even if you're using the blue tray.
Yeah.
Okay.
But if you would need to pull the blue tray out to get your band,
yeah.
Right.
So
you'll have to wear the glasses.
Yeah. So what happens?
Okay.
Yeah. What happens when you pull the blue tray out is and we haven't tried this. It should say turn transilluminator light on. And you can click that, and it will turn the light on, and you can cut your gel, But if you have the other tray on and the UV light is needed to see your samples, because I didn't buy another piece of equipment, you cannot cut your band out off of the stain-free tray because it has a box, kind of a lucite or a UV protective box, so you have to set inside of there, and it clicks in somewhere that the machine recognizes, and so when you have the drawer open, it won't let you turn the UV light unless that is in place.
Mm-hmm.
So I didn't buy that yet. I probably will eventually. It's like $275 or something. But I figured most of the people that want to cut with UV light, again, can put it on there then once they've imaged it and can cut the band out over there. Mm-hmm. This one's a little safer. That one's a full UV light on you, and blocked. And this one, at least, it's a kind of an enclosed box with an open bottom, so it sits on top of the gel, and then you pick up the top of it towards you and put your arms around it and cut, so you sort of have a shield in front of you and around you. Okay.
So what's the idea about cutting on those?
Yeah.
What's going to be the rules?
So
on
cutting.
that?
So look, a glass plate.
Oh.
So it would be nice if you would put this under your gel if you plan on cutting, so you're not scratching.
Scratching the tray.
Yeah, because you'll see once we get the camera live image that you can see every speck of dust on there. You know, so I'm sure if you had scratches on there, that might start showing up
Yeah,
on your
very nice.
gel. But so yeah, I prefer you put your gel on glass and.
They make the gel tips too that you can just put on a pipette.
Plastic kind of punching.
Yep.
Oh, okay, I haven't seen those. Okay. All right.
Yeah, I've seen those.
So, we have 3 gels, it looks like, to image, so let's do that. So, open door, pull out drawer. Not too difficult. Let's start with mine, since I've got it here. Mine, I cannot use the blue tray with. This is Easy Vision dye from Amresco. It comes in a dropper bottle the way I bought it, and this you can see locks in, so there's a little notch here that it fits in, and then underneath there's a little oblong thing that it kind of locks into, and if you don't have it on right, it'll tell you. It complains. Okay. You know, since it's stain free and I can't get gloves on very well, I'm just going to slide my gel on. You do see these bubbles underneath there, so you want to get them out. And you want to pretty much center it. And you can do this a little bit better once everything is live here. Okay, so that's a live image of the gel sitting underneath there. And right now it's basically, the way it's set, it's too large. The gel looks too large for the section that I want to do. So you go here to image size. Well, actually, first thing you do is up here. It's always set on single, so this is a single image versus a multiple image. And we're not doing multi-images unless we're doing chemiluminescence. So you always keep this on single, okay? Image size, you've got 3 major choices: small, medium, and large. And you can see this is the small image. If I touch medium, it actually decreases the size of the gel on the picture, and large it does again. So if you want to go in between here, I think there's a couple different ways to go. First of all, you can enter numbers yourself, and right now you can see medium is 15.5 by 12.4 centimeters. If we want it a little bit larger, you know, I can increase that to like 17. And you can see the range is 9 to 21. And then it'll change the other one. Actually, I have to go the other way. I did the wrong way. It's not obvious. It's backwards. Do you think it is? The lower numbers make it bigger?
Oh.
Yeah. That's
Well, is
15, that's 9. So
We're
let's
going
try.
by the size of the gel, not the size of the image.
I don't know. We haven't figured that out yet.
Okay.
We do know that whatever you see in this frame of a picture here is what you print exactly.
Okay.
So I will get dark spaces here if I print it like it is here. And you can't really, like, you know, you're not going to broaden your gel. It's a narrow gel. It's going to be, that's going to be that way. But this is the best I could do here, because I'm still seeing my wells here, and I can see the bottom of the
That's
gel.
if you turn.
You could turn it.
That would
Yep,
work.
you can
That would
turn
work
it
as
too.
well.
I don't
Okay.
like turn gels in
Yeah.
my pictures, but you might. Not my thing. So the other thing you can do that I figured out, I think from reading, and let's see if this works here. So you can do this and change the size.
Like a.
Oh, you can do that.
Yeah, you
'Cause
can.
we were trying to do that.
We
Yeah.
were trying to do that.
How did you do that?
Like this, with my fingers holding them down.
Just like a cell phone.
Yeah, like a cell phone thing. But it's opposite. Pinching makes it larger. Is that right? And yeah, and that makes it smaller. Isn't it opposite again?
Yeah.
Is it just me?
Maybe there's a setting. For
Yeah. So anyhow, you can do that, but I kind of like what did I have
Was
before?
it just under
12?
image size?
Yeah, you just stay under image size, and you just can use your hands to. Yeah.
Oh, that's much easier than just randomly putting in numbers.
Yeah. I mean, you can randomly put them in. You know, it depends how much you want to mess around with it. And the other thing you can do, because Adrian is like, oh, I want it. You know, it's not quite right. So you can reach your hand in there, and I'll be good and put the glove and get it so that you're more even here. You know, so you can see the top. Oops, I go the wrong way. Here's the top of the gel. That's better.
There you
A
go.
little crooked, but not too bad. So you can mess with it and get it just the way you want it. And so after you've fooled with the image size, and again, yeah, you could turn it sideways, you have to select your application. And so, of course, most of ours will be nucleic acid, so nucleic acid gel's right there. Okay, if you do protein, if you do blots, you can see the other two choices. And then it shows you screens. So obviously this is the blue screen, this is the white one for proteins, and then this is the multi-use one. And so my dye isn't listed on here. So when I first did this, I had to figure out, well, which one do I choose? So I chose gel green. And gel green pretty much always uses this screen. You can see the gel green isn't listed anywhere else here with another screen. And sorry, I shouldn't say that. Mine you can't see with the blue dye. Gel green you can see with both dyes, but you can see it with this green, and it works with it. I don't know, I think I could also have picked maybe cyber gold, and it still would work as well. But you can play around with this because there's a preview image. So
now these choices are the choices. I mean, if something else comes out down the road and we get a different dye or whatever,
you just try one of these.
You just figure out which one of these works. We're not going to try and add it. Or,
No, I don't know that you can add
that's
it.
what
I didn't see
yeah.
anything in the manual that
Okay.
said you could add it.
Okay.
So I think this is what you got.
Maybe
And-
post a little sign if you're using a gel that's not, or a dye that's not on there. Say like, yeah, it worked with this.
Yeah. We could make a chart and say that which screen and which choice here works with it. And so yeah, that's something we just had to play with. I can't remember if we tried gel red or not, but gel green works, I know. Yeah, so you choose that, and then it pretty much knows what light source to use too, because if you're using the blue screen, I'm pretty sure it uses the epi white light, not the UV, because you can keep the drawer open. Okay, so those two are chosen, and then you have an exposure choice. Now you can just leave it here, auto optimal, and you'll get just a fine picture. If you want to fool with it, there's this rapid auto exposure. It doesn't mean that it's quicker to do the exposure. It changes the pixel density a little bit. And we played with that and we didn't really see a lot of difference in it. The other thing you can do, instead of auto, you can do a manual exposure setting. And that's best to be fooled with after you've already taken a picture, and then you can check the properties of the picture. It'll tell you how long it exposed it. If you want it darker or lighter, you can raise or lower the exposure time yourself later, okay? So we're gonna leave it on optimal exposure. So everything is set. Then you can take a preview image and you can see what your image looks like.
Now you said you do use the UV light? for
This
or
one
you
is
don't?
using
You
UV
do,
light.
okay.
Yeah. And it picks its own. When you pick all these other things, it knows what light to use. I mean, okay. So it gives you these little quick help things. I don't know. They don't pop up very often, but if you want to do this, do that. Yeah. So it gives you a box. We're just going to get rid of that. There it is. And it allows you to adjust for optimal exposure of a certain area if you want. So if you have a really faint band that you want to see a little better, you can do that. So we can take it, we can drag the box. These are some digest products of these two PCR reactions. And I'm changing the gel size too. I don't want to do that. What am I doing? Oh, here we go. It's even hard to do this one-handed, guys. Gosh, this one-handed thing is getting really old. Let me tell you. Okay, say I want these bands to be more intense. So I kind of ringed them and left the rest alone. And then I'm going to ask it, see, I think, we open this to get rid of that preview?
I can't remember.
That'll get rid of the but see, then you lose that thing. Oh, let's try that again. I think you just have to take the picture then. I'm going to use it a couple times. I'm still kind of learning myself. You can't really do anything wrong, though. I mean, it's
Right.
like, don't believe.
Is that ethical, gel manipulation?
If it's only for your notes.
It's only for your notes. So now this is actually, so this is just a preview. This is actually going to take the picture.
That's funny. Yeah, you'd think you'd be able to re-preview it.
Yeah, maybe, but it doesn't leave
Doesn't seem-
preview on the screen, so
doesn't
you
seem like-
can't just
Mm.
re-preview it. That's why I was.
And you can't select the bands before you do the preview?
No,
you
it
wouldn't
doesn't
know-
give you the
yeah,
box.
because you wouldn't know where to
Because
put
you
the
don't know
box.
where they are, yeah.
Yeah, that would make sense.
What
Okay.
about the little sun image right next? Does it?
Yeah. So now, because I've increased these are like saturated. So it does change it. And it, depending on if you really want to see those for some reason, you can do that. But this isn't maybe an image you necessarily want to save for.
So it did, when you boxed those lighter bands.
Yes.
So then it increased the intensity for everything.
Yes,
Not
because
just in that
it
box.
increased the exposure
So you
time to
just
make
said-
those darker.
So you're basically telling it, this is what I want to see.
This is what I
So
want to see.
the rest of the gel, you just
Right,
ignore
right.
it. Okay.
So
got
I
you.
mean, I prefer it
So
the
then
other.
that's okay.
Yeah,
As long as it's doing the same thing over the whole.
yeah, it's
The whole
doing the
gel.
same thing over the whole gel.
Okay.
So, you know, some people complain their bands are so faint all the time when they're printing. Well, this is a way to get them more intense.
To get, without monkeying around
Without
forever.
monkeying around
I mean,
in-
you just say, these are
Right.
the ones I want to be able to see, and it'll give you
Right,
the optimum.
right. And because a lot of times the markers are maybe way outshining
Yeah,
your sample band. So
Yeah.
you can box all your sample bands and not the markers, for instance.
Yeah.
And you know, it didn't oversaturate much of the markers, just one of the heavier bands that's
Right.
used as sort of a
Right.
gauge. That's 500 or something.
Okay, that's cool.
Yeah, so this is 100 base pair ladder, and this is the 1 kV plus ladder next to it. So,
actually,
yeah,
Actually, that's a re-preview then, because it didn't really print yet.
well, you have to tell it to print.
Right.
But I mean,
I
if
thought it
this
was
is a
pretty
picture in the gallery right now,
Oh, I see.
so you have created a picture in
I
your
see.
gallery.
Okay.
Filling up the gallery, and actually it's in your gallery because we're in your user. I meant to switch to mine, but that's all right. You can delete them, or we'll delete them at the end. But yeah, so I mean, if you like this, fine. But when you print this out, these are going to be like, whoa. And actually, when you print this like this, and we can do it, just press print. It says the selected image is about to be printed. Yes, okay. And then it's going to print it. These will actually, I think it clears it out to black almost. It's so, yeah, it's so intense
Oh,
that it clears it out to
so
black.
instead of just washing out the whole gel. it
Yeah.
just- okay.
Yeah. Okay. So, I mean, I wouldn't necessarily like that picture for most of the time. I would rather have that first view that I had.
Right.
Because those bands weren't that.
No.
You know.
No.
But that was the best I could do running gel today. I was trying to get some light bands. But so that's one of the options. So what else can we do? So on this picture now, since it's in our gallery now, and we can do some more transformations on it actually. Let's just go across the bottom here. So here's transform. And you can play with it like you did the other one, where you're darkening up the background, or you're, you know, increasing the intensity that way. There's even an up and down here that can be fooled with. Okay, so you can do manipulation there, and save it. You can
Which-
invert the image.
Which manipulation was that? I'm
This
sorry,
is
I missed
this is
Oh,
transform.
okay, gotcha.
Yeah,
Can
you
get
can
rid of the red?
you can what?
Can you get rid of the red?
You can get rid of the red if you probably have to retake that picture because of the way we adjusted. I think you have to retake the picture without that adjustment of that boxing, and let it just do it normally without any intensity differences. Yeah, because I don't think that'll go away at this point. I think, I'm not sure what that does. That's just changing, yeah, just sliding it back and forth by itself. Auto.
Is that the double readings? We're going to target double readings?
These are a test of the primers for DRB, or no, DMR, and both DMR target sites. They're opposite each other, either end. Just regen, undigested product and digested product, 2 different concentrations. So we have that, and you can, you know, manipulate it as much as you want, and then take the picture again. So that's all you can do there, I think, and I hate that inverted image personally.
And those sorts of things, these sorts of adjustments you're doing now, can only be done to the image after you've taken the image, or can you? Because you can't do it before.
I don't think you can do it in the preview. No, you don't get this bar coming out on the preview. So it's in your library now, but it's easy enough. You just press delete here if you don't want it, and you start over again. You don't have, it's not hard to get rid of if you don't like it. And then you also here have the image info. And the image info is the place where now you can change the name of it, because it gave it this ungodly, her username, the date, and the hours and minutes and seconds that it was taken at. And so you can just click on that, bring the keyboard up and change the name of it. And you can also add notes about it. So if you want to make a comment on what it was exactly, or say it's in page 43 of my notebook, and you can go back and, and correlate it to that. Yeah, so you can do that. Let's see, is there anything else? Oh, then the other thing is, it tells you what application you picked to take it. So you're on gel green here. You use UV transillumination and the filter it used. And then it also tells you the, let's see, where is that? Exposure time, 2.879 seconds. And so again, if you want to change that exposure time on the manual setting earlier, you could bring it up or down to make it darker or lighter.
And that information is saved with the image?
With the photo, yeah.
And so when you pull up the image,
Out of the gallery, you can find that
you
again.
can find that information again.
So this gallery connects to your smartphone?
Yeah, take it out with the
That's
USB.
the next version.
Yeah. Yeah. My daughter did buy a new camera that will download pictures straight to her smartphone just by putting them near each other. That was cool. Like
You just
that.
take a picture and it's in your cell phone.
Yeah. Okay, so I mean, it gives you coordinates and pixel ranges. The other thing you can do, and I haven't tried to do this yet, is if you touch the screen there, it gives you a saturation number. So like if you touch here, it gives you 100% saturation because it's red. It's like 100%, but if you touch here, it's 47% saturated. So you can get ideas of band intensities if you want to do it that way as well. And then it just disappears. Yeah, let's see. What else? Okay, I think that's most of what you can do there. This merge you won't be using unless you're taking multi shots and chemiluminescent stuff and like layering maybe a stained blot on top of the chemiluminescent image. So you won't be doing that with this. There's the print, there's the delete, and then there's send save. So we can press that right now. Send save, you have export options. You have as an ImageLab file, which is one you can manipulate with the ImageLab software to like calculate molecular weights and label and things like that. And I think, I don't know the difference between this ImageLab option and the analysis option. I mean, obviously, I think you probably have to download the analysis option to do all those things. The ImageLab itself option, I'm not really sure. I don't remember them defining that in the handbook. I'll have to look at that. You can also do TIFF and JPEG options, and in order for this USB to come up, you have to have one in there. Somewhere. Not. There. And now it's asking you if you want to save it to the USB. So we can just press that and it'll save it to your USB. And then eventually we can save it over here when I figure that out.
What format will it save it in if you put it in USB?
Whatever you want. JPEG,
Oh, okay, that's
TIFF.
what it is.
It'll give you
Yeah,
multiple
because the
ones.
ImageLab, I guess we won't be able to open it.
The ImageLab, you can open it in here
Right.
on the image software, ImageLab software.
Or you can get the software,
Oops.
or
Or-
if we get the software from you,
Yeah, I have the software on
Oh,
here. If you want to load it
she has
onto your computer, if you want to do something with it, you can certainly. It was free to download, so. But it is on here, and so you can put your USB in here. But I'm trying to develop a shared folder over here that it will transfer to instead of just to that. I haven't quite figured that out. Okay, so that's pretty easy. Let's see. Next. Let's see. That we don't need to fool with. Let's go to Well, let me show you the gallery here. The gallery here is going to be kind of boring because we only took one picture, right? So you see the one picture. This is the gallery. And it'll show you thumbnails. You can make the thumbnails larger or smaller by doing this. But if you really want to open it up full screen, you just have to touch it. Sorry, double tap it. And it will open up full screen again and let you do whatever again with it. You can transform it still. You can do all sorts of stuff with it. Okay. If we went, let's do this. Let's go out of her. Well, actually, let's just stay here since we're in here. Let's image your gel, and we'll put a couple more pictures in her gallery, and then I'll show you that you can look at more than one picture out of the gallery at once and compare them on the screen together. Okay, let's do that. Let's try some other dyes here.
Okay, that off.
Okay. So your stain, we'll put on this one first. Do you know if it can be used with a transilluminator?
It's supposed to be. That's why I bought it.
The blue one?
The blue one.
Okay, let's try. Let's put it on there, and then we'll put on the blue.
Yeah,
one.
because
Let's
I
see
think
what
the
it looks like.
dye. I think it's supposed. The green dye is supposed to work on both.
Okay. Yeah, I think so too. Yeah. Okay.
It'd be interesting to see the difference.
Yeah. And we can compare them side by side. We'll take pictures of both. How's that? Okay.
Did you guys ever try to compare between deuterium bromide, like saying amount
Well,
of DNA and
if you look at all these pamphlets, they do that,
Okay.
if you believe them. Supposedly, like I said, the cyber safe dye seems to be not maybe quite as sensitive, but that was like the first, like the forerunner of the safe dyes. And now there's this whole other group of safe dyes that are purported to be actually better. The fluorescence is higher than Ethidium. And you could see my ladder perfectly well. It was a very low amount of ladder. You can see the 100 base pair thing. The only thing about these dyes, the other thing that's different about them is mobility shifts. So you told me the other day that you thought the gel red,
For larger pieces of fragments, and I'm wondering too if it's also maybe fragments with like superstructure.
okay,
The dye gets caught in there and it slows down. I should have saved my gel from yesterday because it has. You can see the ladder looks pretty good, but I have a 669 and 700 base pair ladder, and they're both like crammed up by around 1100. So
Okay.
it just slows down the migration.
okay. And for some of those dyes, the recommendation is not to do in-gel staining,
Post-stain.
post-stain.
To do a posting.
Yeah, to do a post-stain, because they do fine there, it's just that when it's in the gel, it restricts the mobility. But that takes some extra time. So do that. Okay, so to get back to the camera, we just press camera there, and we should get another live shot. And now we'll have to adjust the image size. You want to do this? Since you got Go on, come on.
I'll watch.
Oh, geez. It's supposed to be easy, you see? So,
Did you do your staining with your gel in the gel? Or did you do it post running?
in gel.
You did, okay.
Yeah, mine doesn't seem to inhibit the bands at all. Everything looked.
It depends. Yeah, the smaller stuff you
Yeah,
don't notice.
no,
it at all.
and the
Sure, it's gonna
pamphlets do
depend
mention
on
that.
what charge they
Actually,
have.
quite a few look like they inhibit.
the safe dyes are larger, and that's why like it doesn't get into your skin and doesn't get into your DNA.
It doesn't get into
Bigger.
cells. It doesn't
Yeah.
get through gloves.
It
Yeah,
doesn't go through
exactly.
the gel.
Okay. So, I mean, that's a pretty good size. You know, we could play around with it a little bit to make it pretty big,
I
but
just found whatever DNA I had on hand, so
yeah.
I don't even know what it's gonna look like.
Okay, so this is gel red.
Yeah, it's still red.
Okay, so we actually have a choice this time, gel red. And we'll do auto optimal. We'll just leave that, and let's take a preview. It's doing it.
It's gonna look terrible now.
Have you looked at this yet?
No. Oh wow.
Didn't run very far. It's good.
No. But like that, the Midori green. Oh, wait. Oh yeah, the Midori green looks.
So which is what?
I'm just like,
here?
hold on. I'm like,
Is the gel full, or is it just those 4 lanes? Or
Is the Midori green over here?
It is. You're right.
So
Thank
we're not
you.
seeing it?
Yeah.
Yeah.
So it doesn't show up on here, or doesn't show up enough on here.
Okay, yeah.
This was a regular dose, and then I just had a little bit more in the tube, so I added it to the last one just to see if. So that is like probably
These
on the
are
left.
ladders?
The left is like probably 1 microliter of ladder of mixed ladder.
Okay, I mean, it's a
the
nice,
gel green or
No,
gel red.
Gel red. Yep.
Gel red.
but so you're not seeing the other
Yep.
dye
I'm
at
not
all?
seeing the other one. That I was like, yeah. I'm
We'll
like,
have to try a different
We'll try,
combination.
we'll try the blue screen to
Yeah.
see if we
Yeah.
see it, otherwise it maybe is crapped out.
Yeah.
Because I think, well, I don't know if you can see all of them under Ethidium. Maybe some of them just under trans UV or trans blue.
I thought it said you could, but
Okay,
maybe it's a
okay.
sensitivity thing where
Okay.
like you maybe.
So
can't.
because this is such a little area that you want, you know, you could move the gel down and make it bigger and take a much bigger photograph too.
I have another meeting.
Okay. So, you know, you can do that too. And, and get a much nicer photograph, you know, if you really wanted to, since you know you don't have much there. You'll have to kind of guesstimate a little bit on doing that, because once you go live again, you'll lose, once you open this, you lose the image.
Yeah.
But yeah, you could do
What
that.
is this little thing do?
Oh, that
sorry.
little thing?
Yeah.
Just a minute, I'm going to move this down.
I think it was just a little circle.
Yeah, I was wondering if you like highlight the
Maybe-
bands, maybe you can
Let me see.
do exposures.
I don't know. I know what that circle is. We'll see. Okay, see, that's better. Yes. So this thing, I think that just indicates that you can brighten things up with this box, maybe, rimming it. But in this case, you know, the only thing you wouldn't do anything with this
Yeah,
one.
it's
I mean,
fine.
it's just fine. So, so we can, let's take a shot of that. We won't print it, but we'll just take a shot. So we have another one in the gallery, so we can fool with them later. And then let's try the other tray.
Yeah, I want to see if we can reuse.
Okay, open the drawer. You can do that. Pull out the tray. Why don't you put the gel in there first? And then you can... It's a mega gel. Daddy. Exactly where I had that situated. Get the giant bubble going there. Come out.
So did you ever figure out that
Yeah, get that.
does the gel doc know which tray is in? Does it have some kind of sensor?
Yeah, it knows which tray is in.
Okay.
So we'll go back to camera. And so we're gonna pick something with a blue screen.
Probably the gel green
Yeah, let's pick
or
that.
sour green, whichever.
And then we'll preview it and see if we see it. And I've got my screen all mucked up here, right? Huh? Didn't press it. Look,
Yeah,
you
something
can see it a
still
little
there.
bit more.
It's magic.
It does make a difference.
Yeah.
Are they the same DNA, the same?
It was the same plate. It was just a old plate I found in the freezer that hadn't been run. So
Okay. Okay.
Because I mean, these do appear to have run a little further.
yeah, and that's the thing too. That's what I wanted to see. I wanted to see, and
We can enhance
wanted-
that. Should we
yeah,
enhance it?
well, I wanted to compare the two ladders next to each other too to see if the Midori green
Yeah.
had less drag, and of course, I should have done that with the horrible marker, but I didn't do it.
Next.
Yeah.
Let's see this. See if we can.
And it might be really great. It might just be like saying, "Oh well, we have all this beautiful one."
See how much you can actually do with those light bands.
That's really fine.
Yeah, the exposure time's long. Looks like.
Because it's more sensitive.
Yeah.
So it's safe to get like the whole tray apart especially I'm seeing fingerprints over here, to get the whole
Yeah,
tray
I think
wet.
so. I don't, there's nothing, it's plastic and then the,
Just say not dishwasher safe.
yeah, yeah.
It does say that?
Yeah, it does.
Okay.
Don't put it in your dishwasher.
What about microwave? Can I microwave it?
No.
Remelt my gel on it?
Yeah, wow, that's a long time. Yeah, look at it.
Yeah.
Ah, 46 seconds. These other things are going to be like, well, screaming memes, like, yeah.
This is really cool.
I'm just hoping it's just because it's old.
This would be
New
so
job.
good for people to do Western.
Oh, yeah. Yeah.
Yeah.
You wanna
Yeah.
quantify bands or
Oh, and the chemiluminescence thing too. You set it up for exposures, and every like 5 seconds you can say take a picture.
Yeah.
You know, so you have a whole series of them, and then you can pick the one that's best, and then you take it and you analyze the intensity of the luminescence with the software. Yeah. And then if you've stained that on top of it with something that you can have a visual stain on, you can overlay them on each other. Wow. We're asking it to do a lot here.
We're
Holy
maxing it
crap.
out.
There.
Oh,
But look at they are a lot better though.
Yeah.
If you really were desperate to see some bands, you know.
Are these markers the same?
It's the same ladder, yeah.
Set side by side?
Just
So this band is this
Yeah.
band here.
So it does look, and like these are really
Crunched. Yeah.
compressed.
Yeah, yeah. So the dyes do matter that you pick. And you know, maybe you want a couple different ones to use, you know.
Yeah,
if
or
you-
find one that you like. I
Or
guess.
find one that
I
you
mean,
like. Now, the one that you use that we'll do next, he got from Storehouse, from Newmarket. And so that might be one of the easier ones to obtain if you just order through Newmarket, and probably a reasonably priced one. My stain that I was using, I think I bought it from like VWR or something, and it came in a dropper bottle, and they said use 1 to 2 drops per 50 ml gel. Well, the first time I used 1 drop, and it stained plenty good, and there was almost like background on it. And then the second time I ran 100 ml gel and used half that much because I measured kind of what a drop was, you know. This time I used like a quarter of that, and it's still staining just fine. So don't necessarily go by the recommendations because these things are expensive. And so if you can use a quarter or a fifth of what they say, then do that. Especially because, like I said, I thought I saw a lot more whitish background, and I'd rather not have that.
probably
You
get a
know.
better image.
Now, some of these stains do behave like ethidium, where it runs opposite the way the DNA runs, and some don't. So that's another thing you'll see is maybe you'll see that wall of stain going up, and maybe it'll just be. I'd rather see it just across the whole background even, but this one seems to be fairly even. And all these specks you see, I should mention this, are dirt on
Yeah.
glass and stuff. It's really sensitive. So if you really want a clean looking picture, you need to clean that glass off and clean the dust off of there before you use it, because it's sensitive. So yeah, I mean, okay, that's cool.
So
All
can
right.
can you print this one?
Yeah.
Use it.
Well, I want to take it down. I want to measure the distance it's traveled.
Okay, so yeah, the image info on this. So yeah, 46-second exposure compared to like 2 to 3 second on
Yeah.
that first one. So it really tries to see your stuff.
Although, does it, for like, it gave you different options. Like it gave you the cyber green and it gave you like a different green. Is it slightly different the wavelength that it picks up on?
It
different
might.
ones?
We can try. Let's go back. So that's cyber green.
Try gel green.
Gel green.
Top one.
Up right in the middle.
Gel green. There. Okay,
We have the
let's
right
try
color.
that.
Yeah, we do. Yeah.
See, will that box stay there though? Did you want to see this light stuff?
I just want to see if it's any different.
Oh, okay. This won't be the long exposure because we didn't change it. But we did see that first one, and it was. Or did it keep?
I think it did.
It did. It
Oh,
must have
next-
because we didn't open this.
No.
So it
Thanks.
probably kept the box the same way. I know to erase that you can just open the door and it takes everything off. So yeah, no, it's obviously doing it for a long time again. Oh, maybe not.
Yeah, not,
No, maybe not.
I mean, less
It-
less
it-
background, but
It is
Well,
less.
but yeah, but But the only reason there's a lot of background on this one is we had to have it so long.
Right.
Yeah.
If we extended this one, it would look just
That's
the same.
true.
I think.
Right, and yeah, and I
right.
think those bands look the same intensity as that very first one that we saw.
The very
Can
first
you
one we
print this
didn't
one?
see it at all.
Well, that was on the other tray,
Can you print this one too?
on this tray. And well, I'll just say that this thermal paper isn't cheap, as usual.
Let
Not that
people
I'm
know if we
scolding
let me
you.
know if we have to buy some.
Yeah, but the other option here now that we didn't have maybe as much as, if you take the image off and just want to print it on your computer printer, you can do that too.
So you don't have to do the.
You don't have to do the photo printer here, no, because you can just print a JPEG file on your computer. Now, usually though, because that's what we do in Mindy's lab, because she doesn't have a printer on her, she has a little mini gel dock kind of, you can't see bands like that. You know, but you hardly do here. So if we go back to the gallery now you've got more images. I'm going to reduce the size so we can see smaller thumbnails. And if you want to compare all these together, if you took like exact images and just fooled, you can check all of them. Whoops, I didn't do that right. I double tapped it, just one tap, and activate all those. And so when there's a blue line around it, that means it's a selected image. So there's blue lines around all these, and then you go to view 4 selected images, and it's going to show you them side by side
Side by
here.
side, no, that's
Or you can do 2 or 3, up to 4. So you can look at them that way as well. You can also, in that view, if you took some today, you took some yesterday, if you want to see all of yesterday's, you click the date on that, like this date is clicked right now, and it will show you all those images from yesterday. Or if you have you've named it, some DMR6, and you want to see all the DMR6 images, it'll bring all those up into one area that you can see them all. But the bigger screen will only show you 4 at once.
Now, you said before that it's saved somewhere with this image. It's saving the exposure time and lighting, all
Yeah.
that. Now, if you let's say you came into your gallery and you're taking pictures on different days and you want to compare them, but you want to make sure that the exposure is the same. How do you see that? How do you see what the exposure time and?
You just have to click on, double tap one, and then go back into image
Oh, okay,
info.
okay, gotcha.
And it'll tell you.
Okay,
It'll
cool.
tell you the exposure time.
Just to make sure that
Yeah,
you're comparing
there is
apples to
something.
apples.
There is something. I think it's for the luminescence stuff, maybe, where you can actually make everything the same. There's some. I can't remember which one of the properties, but yeah, you can do something that equalize things. But it's kind of nice sometimes to compare 2 images side by side. I don't think. I don't know if it prints it. I don't know if there's a print option in that.
So the search button on that side, is that only the file name search, or does it also search notes?
I don't know if it'll search notes. It'll search. Yeah, it
Well,
searches.
can we add a note in and see
Yeah,
if it
we
searches
could.
for it?
Yeah. Let's see. Image info.
Midori.
M
It's like the
M
blues.
I D O R I. Yes.
Yeah.
Okay. All right, and then we'll go back to gallery and we'll say.
Oh yeah, it is searching.
Is it going?
Yep.
Yeah, I did it.
They found it.
Oh,
yeah.
you found it, yeah.
Oops. Yeah, I did find it. I just have to get that off.
Get
You
that
just do
done.
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 Oh, you're right. Done. Thank
There we
you.
go. Oh,
And
yeah.
it did find it.
Nice.
Yeah. So, and then in order to get this off and go farther, you have to actually wipe this out, I guess. Otherwise, it keeps showing you that. But, yeah. So, and then you can also delete images out of here. You just activate it with the one tap, and then you delete from there. Okay, and you can also still print and stuff from there too. If you select an image and you hadn't printed it yet, you can bring it up and then print it.
I had a question I was going to mention and I got distracted. But when you were doing the manipulations, so after you had the image, and then you were doing the manipulations with the light and dark and
Yeah.
everything, and you said if you don't like what you see, you can just delete it. when you press the delete when you're doing that, it's just deleting the manipulations you did, not the original.
No, the delete will delete the whole file. That deletes down here, deletes the whole file.
Okay.
You can put it back. I think there's an auto button, and you let it go back to auto if you wanted it the way it started.
To go to just get rid of whatever you've
You
done.
tried, yeah.
You want to try
Yeah, I
something
think you
else.
can just press auto next to
Okay.
that. We'll look at that on this next
Yeah,
image
okay.
here.
Okay.
Now, when you export it, does it export the metadata with it, or is that it's only on the computer end?
The metadata.
So, like all the exposure, the notes,
Oh,
all that kind of stuff.
you know, I'm not sure. I doubt it. I think it's just the image.
Okay.
Well, JPEG files have a lot of
Yeah,
data
if you
attached
go
to
to-
them from your camera.
Oh, maybe.
Yeah,
Well,
'cause-
maybe. You know, I think you will get that metadata if you take it out as the actual image file from the software.
It's native image file.
But yeah, the
Okay.
only way you can view that type of image, though, I think, is on the software, on the image touch software, or the image lab.
Could you
software
add
itself.
notes from the computer from a separate source, or is it only on the computer? It seemed like it would get kind of old typing in on the touchscreen after a while.
I don't know. I haven't tried that.
Okay, something I can
They
try.
didn't mention that in the manual.
Okay, thank you.
Yeah. Did you do this? Well, this is the blue screen, and see, his die works with both screens. Mine does not. Mine does not work with the blue screen.
So those are right now. Those are the only two screens options we have, right?
Yeah, for the DNA gels, that's all
And
you
that's-
need. And then
that's-
this
yeah,
is for the
that's
protein and
protein
stuff.
stuff.
So for now, I think we're just going to lay them on there. You can buy a $77 holder for these, plus it'll hold that protective UV light box thing for over the gels to cut. For like $77. It's like I was trying to find a test tube rack, a rack that had a wide enough girth to like hold it, and I didn't have any in my lab that would go down between the pegs or something. So if anyone has one,
Or go to Target and get it.
or go to Target, yeah, that's what I thought too. And I thought that exactly. Dish drying rack.
So the one that's boxed, is that from the last photo?
That was from the last photo.
Is that what you did?
And you know, they say that the applications that are recommended are the asterisked ones, but I haven't figured that out yet.
I guess it's all the blues.
Yeah, it's all the.
So I just have to pick between the blues.
Yeah,
then.
you have to pick between the blues.
I guess, yeah, I guess I don't really
Because
know.
yours is called.
I don't know, it's some generic
Um,
DNA save.
I have it here. Yeah, it's a weird one too. Mine's weird too. Safe DNA gel stain. Apex. Safe DNA pre-stain by Apex Bio
They couldn't
Research
come up with
Products.
a more clever name.
Yeah, I mean,
as
that's
good
pretty
as they could
generic.
do.
I
Well,
know.
we'll try CyberSafe and see what. So I mean, I don't have to do these other ones, right? Like the image size?
Well, no, not if it looks right now.
Right.
Yeah, then you're good.
Okay.
And then you just compose that and preview it.
Oh, yeah.
I mean, I guess you don't have to preview it. I haven't tried just going right to the
Oh,
camera.
yeah. So I was thinking this is kind of a lot longer process than, you know, if you just
Yeah, I
want to
don't,
like, come in.
let's try that
snap
after
a
this.
photo
Let's
and-
just see if, you can just go straight to the camera. Whoa, look at all
Oh,
them
wow, you
bands.
got
It's funny because these are all different concentrations of ladder.
The same ladder?
No.
Two different ladders, right?
Mhm.
Okay.
But it gets, you know, from the most, 10 microliters of ladder, 5, 2.5. So even the 2.5 shows up.
Pretty bright, yeah.
Yeah,
Yeah, it's pretty, that's nice.
Yeah.
Yeah. You can use a lot less ladder, and, I mean, those are expensive too. Okay.
So let's say I just want to save, I guess saving it.
And did you stain it in the gel as well, or did you post?
No, in the
Yeah,
gel.
Okay.
Yeah, most of these that you can do in gel, you can add them to a hot gel like you can at Vidium, and it doesn't warp them or do anything weird, but there are mobility
Yeah, I was
shifts
gonna say,
mentioned in a lot of them.
If people are worried about the mobility of these dyes, then maybe.
But you can post-stain,
Yeah.
and those post-stain solutions, generally you can save for other, for a couple more gels.
Mhm.
Yeah. But you probably would use more of it if you had to put a whole bath of it in, depending on the gel
and
size.
you end up making bigger messes and everything else.
You're probably using more too if you
Yeah, you're
post
probably
it.
using more.
Yeah.
I mean, on this gel of mine, I use 10 microliters of this stuff.
It just might be a consideration.
And I think they said it would do 300 gel.
It just might be a consideration when you decide which gel to use. to try and figure out which one's kind of right for your application as far as mobility.
For most of our stuff, even if it slows down the migration, I'm like, it might not be, the bands might not be where you think they're supposed to be, but they're still separated the right way.
And if you're running a size marker,
Yeah, well, I'm
They
like,
should-
Are the size markers doing the same thing though?
no, they don't. That's the thing. I'm like, this
Oh.
size marker. That's why I'm wondering if it has something to do with the complexity of
The DNA.
the DNA superstructure,
Yeah, yeah,
because
yeah.
the latter isn't as affected. You'd think
Oh.
that the upper ones would be more affected, but I don't think it is.
Okay, okay.
I will
So
let
it's
you know,
not-
but I'm like, I think that obviously there's a difference between the Midori green and the gel red.
Yeah,
so
yeah, definitely.
so it's not
Yeah.
just strictly a size.
I don't think it's strictly a
And
size.
I think at
something
least 2
else
or
going
3
on
of
there.
these stain sheets that I copied off do pretty directly say we really don't think you should put it in the gel.
But
they
like,
still
if you know you're getting 2 bands, it's going to be one of the 2 bands.
Right.
You don't. I'm like, you don't even really need your ladder.
Depends on what your application is or whatever.
Yeah, yeah.
So, yeah.
So will it take a picture without previewing? Look.
Should I
Oh,
open
that's already, yeah, you have to start over. Yeah, just open that,
There must be a way to get back to it without opening that.
Yeah, you'd think. You'd think it'd be sophisticated enough.
okay. There were a few options, but this was the easiest one. You can log out, or you can turn the instrument off. I think those are the other 2 options.
So so if I'm happy with all that, then I would just
Yeah.
push this, right? And then I get,
Yeah, and it
and then I
looks
get
like
my
you'll
band.
just-
So like, yeah, if you had a bunch of gels from the same thing.
Once you know that you've got the system that works for your stuff, then you can
Yeah,
just,
you don't have to preview.
so what
it.
did you just press?
Just press camera.
Camera?
Right.
Oh. Yeah, same.
Yeah. So I guess
Okay,
you just
so you
type-
don't have to do the preview.
right.
And then you just tell it to print it.
Right.
Yeah.
I think also because this one is you're looking at the image in that system, it's essentially it's mirroring whatever it's seeing. So that's why you can see it immediately as soon as you shove a gel in there, you can see it.
Yeah, yeah, it's a live
it's a
image
live
right
feed,
away. Right, right. Yeah. So yeah, so I mean, this is really all you have to know. There are a few weird little color cue things. We didn't see any of these, but for instance, if you don't have the tray on there right, it'll tell you that. And sometimes if you don't have some other things right, down along here there'll be a like an orange or a red bar with an exclamation point in it. So you'd want to touch that, and it'll tell you what's wrong. A red one is bad. It's a fatal error, of course, and you have to actually log out and shut down the instrument. The orange ones is a fixable thing, like, I don't know, you've selected the wrong tray type for the one you have in, and it can't go on and stuff like that. So there are a few, and then the blue again rimmed around the picture means it's active. Also, these are green, and so that means they're active right now. So you could either go into the camera or the gallery from this stage here. Yeah, I mean,
I think that's.
And as far as, usability, or I don't know what the right term is. I mean, you just start playing with it. I mean, you're fine with that. I mean, you can't really mess
well,
it
you
up.
can't really hurt anything.
Yeah.
The only thing I think I want people
Because
to
it's not going to do anything that it can't do.
right, no.
And
And
all
it
you-
won't
yeah.
keep any of the lights on when you turn this on. I think if you want to cut from here, let's see, trying to think here. Have I seen this?
Oh, you're to turn
Here.
The light, to turn
So
the
yeah,
light.
so here, so at this point we have the blue screen on. It knows that. And if you wanted to cut the band out from here, you can turn the transilluminator on, and you can see it lit up. Okay, and now if you had orange glasses on, you could
Right.
see your DNA bands and cut them
Yeah, do
out.
you have your orange glasses? Like you know where those are? I want to see
I
those.
feel like
bands.
I've seen some, but I don't know where it
But
is.
yeah, so we
I'll
can't
look
see
around and
obviously
see.
anything. They're supposed to be sending me some. I got to remind them again. This was
But
last
this
week.
is like the UV is not.
The night UV is not on. This is the epi white light,
Right.
I believe. So, but it won't let you do this with the other tray, and it won't turn the UV. You won't have this choice there. It won't do it. Okay. But then the other thing I wanted to say about this was that I prefer people not clean this on here because I don't want to pressing down on here because I don't want these things warped out at all. I don't know how sturdy they are, but you know, there's really no reason to. It's easy to lift off and clean, and then you know, if you did wash it on here, you'd have more chance of like spilling stuff.
Now, with the and the dust particles, that's not. I mean, dust particles on here is not what we're seeing.
I don't know if we're seeing some of both.
It's probably dust particles on that.
It could be some of both.
I mean, if you, I mean,
Yeah.
are you okay with people just like maybe a damp
Yeah,
towel
I
or
don't
something?
think they
Holding it
didn't. You know,
because
they
I feel
didn't
like-
mention anything about
Like, this part of the plastic
scratchability
is
or
really thin. I
anything
don't-
of
I
these
didn't like
things.
pushing
They
down
didn't
up
yeah,
here.
yeah.
That's
Talk
why I
about
put it up
that?
against
Maybe
my.
we need like a soft cloth.
Yeah,
But
I mean,
I think there's enough of a lip here. You could set it on a flat surface to dry it. I
Yeah.
just said I didn't like holding it by the edge and putting pressure on it too.
yeah.
To
Well,
wipe.
the reason I put this cloth on, and you know, this is a fairly non-innocuous thing. You could just stand it right up here. That's what we do at Mindy's lab. She has a tray.
Oh, so you don't even really dry it?
And no, and I don't even dry it, and I just do this, and it dries. So I think I could do that, but this is for the cutting, and if you don't want to use sink water, there's water here to do the washing with, and just squirt it into the sink then. And then to log out, it's just a matter of touching your login thing, and you can log out. Confirm log out, yes. And you're back to the start screen, and then you can just flip it off when you're done. Okay? Pretty easy.
Cool.
Fun.